Jean Fontaine. He's going to give a talk about machine learning-based quality control for NGS data. And I think you have to unmute yourself, Jean.
Hello, can you hear me?
Yeah, c'est bon.
Can you see the screen?
Yes.
Okay, let's go. So thank you for the introduction. Today I'm talking about this quality control of the NGS data. Okay, so the presentation has a really classical structure, no surprise. And I will start introducing DNA sequencing for functional genomics assays and quality control. So the DNA sequencing for functional genomics assays is a complex process depicted here in many steps. And today, it's important I will talk about only functional genomic assay like RNA-seq, ChIP-seq, or DNA-seq, and not and not on full genome sequencing, although we are using the same technology. And there are many question marks because a problem may occur at any step of this long and complicated protocol. And as a user, as an analyst, you will ask yourself, is the data of good or bad quality? And again, by quality, I'm talking about something went wrong either in the technological part or the biological sample is not right. And so are there tools for this quality control? And the idea is that if you keep low quality data, biological samples in your data, you will not produce optimal results. So there are tools to do the quality control, such as this very popular FastQC tool. We can see plots on the left side here, for example, and this will work on the raw data. The raw data are this set of short DNA sequences from the machine. And we will get, for example, here, information on each nucleotide, but we could also have more information on the top right of this slide here, using another tool, Bowtie2, that will map the short sequences to the reference genome and give you statistics. And here, for example, we compare different samples. Each bar is for a sample, but the problem is that it's not easy to know if it makes sense. If this, for example, this second sample is good or bad, there are less reads, for example, but is it too few? You don't know. So you need a very specific knowledge or guidelines. You may also derive other types of quality features. Here we're using the PICAT tool. And it is the same. It's not easy to know if you want to keep all the samples or if you may consider one as an outlier. So other guidelines, like public guidelines, you may refer to. Yes, there are, for example, the ENCODE project provides guidelines. They are very popular. So it's a set of public, so it's a public, of course, and a set of guidelines for best practice in the wet lab. And out of this information, as the analyst, the bioinformatician, as you are, or the computational biologist, you may extract such a sentence, like the minimum acceptable depth for delineation of DHS, so DNA-seq, is 20 million uniquely mapping rates. So here we see. And so now if the plots here are showing an analysis of mine. So I took just a set of high quality files on the left side and a set of low quality side from the same database, from ENCODE. And I'm comparing this 20 million reads, this is this orange line, where it stands in the distribution of values related to this uniquely mapping reads. And we see that we cannot use it to differentiate these high and low quality files. So maybe it's a good minimum to achieve, but here as a practical tool, it is not optimal. Are there better features and can machine learning do better. This is the main point of this work. So next step, I will talk about the methods very shortly to talk about the data source and the machine learning procedure. So the data source is encoded. Why? Because they have manually annotated files for the quality. And we were able to find a good amount of files that are considered revoked in this database, and we consider them as low quality. And we took the same amount of released files from the database that we consider high quality, only for functional genomic assay, ChIP-seq, DNA-seq, and RNA-seq in human and mouse. So on the right side, you see the figures. And in this presentation, we will focus on the data subsets here where we have the most files, because machine learning needs many files. The machine learning procedure is as follows. So each of these files come in this FASTQ file format, the first step here. And we are using 4 different bioinformatics tools, FastQC, Bowtie2, and 2 OR package to derive the quality features. So for example, for FastQC, we have this box to represent a set of features, different analyses, giving information about the quality, and RAWs because they are related to the raw data file. For Bowtie, we will use the mapping statistics. I call it map. The lock, it's a different numbers showing where the sequencing reads or mapping in the genome, maybe in intron, exons, promoters, something like that. And the last set of features is related to the transcription start sites and where the reads map close to it or far away from it. And then with this data, using these quality features, we want to classify the quality of the files, and to generate the best models, we have used in the training procedure a grid search using 10 different machine learning algorithms that are listed on the right side. For each algorithm, we used different sets of parameters to compare and get the best one. And also we applied chi-square or also recursive feature elimination, feature selection techniques. So for a total of 4.6 million models that were trained and validated in this step to produce the optimal classification model. And now we are comparing the results of these optimal models. So in the results section, I will start first to show you what you can achieve without any machine learning, and then we will detail machine learning. So this is results of each feature listed here in these 2 tables. So this number is an area under ROC curve using the values of these features to classify the quality of each file. And so this number at 0.5 is telling you it's random classification, it's not working. And at 1, it's working perfectly. So, and we have divided different columns because we were interested in different data subsets, except the first column is about the whole data. This generates everything all together, ChIP-seq, DNA-seq, and RNA-seq. And so we see that for the whole data altogether, there is no one single feature that performs very good. But here you see already the complexity of this is that you have to eventually pick the right feature for the right situation. And so this is at this level, but one may go at more defined level of subsetting the data, because maybe you may find something related and dependent on the same time. So is machine learning better? Because machine learning will try to combine the features and try to give you the best results. So here we compare just one data. This is a generic data, all the samples all together. The blue bars are the machine learning, the green bars are the no machine learning. And it's always better with machine learning, especially when you use all features. And here this is a result if you limit your models to only the raw features using this FastQC tool. So, and I'm not showing that here, but out of the 10 classification algorithms, there were, especially the decision trees algorithm were performing better, but also I could include multi-layer perceptron. But overall, we chose the random forest because it had basically the best results more often. So does it work with specialized models? Specialized models mean when they are trained only on the subset of the data. And this subset, because maybe this is, for example, here, RNA-seq human. So here on the top left, we see what we have just seen before. And the other panel shows results for different subsets. It is basically the same information. So the machine learning is always better, and it's always better to use all the features. And so out of this, we were focusing all the rest of this analysis to the generic model because it's kind of convenient to have a single model for everything. And yeah, a question was, because here so far, this is global validation or checking the result globally, but how does it work on replicate samples? Let's say you're one person, you're a biologist and you have a single experiment. You're interested to see if it's working in your few samples. So here, this is one experiment extracted from ENCODE, and the points and the crosses are files, NGS files. And the color is because of this manual annotation in ENCODE. And here, the x-axis is actually the predictive probability, the output of our model. And here we see this working perfectly. So yeah, can we see more examples? Because I have picked a very good one. So this is all files related to chip-seq mouse I'm showing here. And in the article, you will see for the other subsets. Yes, basically it's working very well, not perfectly, but often what was interesting is like in this blue rectangle, the order is preserved, even if the red crosses are not exactly at the very right on this plot. So basically this is working. And a very big question is next, does it generalize to external data? External data is data that is not in ENCODE because we are training from data in ENCODE, even if we train the model on ENCODE data and we test on ENCODE data that was not used in training, okay, that's normal. But still, because it's all in the same database, maybe there is a bias. So we took data from the Systrom database that contains chip-like data sets. And we took 610 samples from 32 different data sets. And differently from ENCODE, they are not using like a binary answer, yes, no, good, bad, quality. They're using different flags, quality flags. And so what we did, we compared for each file, the accumulation of bad quality files, this is basically here the x-axis, to this probability that is the output of our model. And we see a very nice correlation. So it's correlating very well everywhere. And we were very happy because it's also correlating to different extent to in ataxic. It's a type of data that is that we did not use at all in the ENCODE, in the training. But what about RNA-seq? Because this Systrom is not having RNA-seq data. So for that, we went to another database, a GeoRepository, for any type of biological data and without any specific quality control procedure. So we took 90 samples from 6 data sets. And what we did for each data set, we generated, like on here on the left side, a PCA, principal component analysis, with all the samples. Then we run our tool that is called the SeqScorer, by the way, to identify 2 samples with the highest probability of low quality in each group. Each group, what are the groups? Each data set is related to one disease and includes biological samples for the disease and a control group. Here, the 2. And so we removed these 4 samples just automatically. We had no idea if they were good or bad. And we then compute again the principal component analysis now on the right side. So after, and we see it's much easier to cluster, to differentiate the data using the gene expression. So it was very a good idea to remove these samples. And I will show you, and this is also highlighted by this Dune index that is just a computation to show the clustering quality. And if I do a difference after and before, if this difference of Dune index is positive, basically I have improved my clustering. And so for the other data sets, so the first one is the data set we have just seen, and the other one, all the data sets that were also included in the paper, we have always an improvement or maybe no improvement, but at least it was never detrimental to the clustering. So this is for us also a very good validation. So in conclusion, without any machine learning, there is a specificity of the quality features to different data subsets, so you should be very careful. With the machine learning, it's better than no machine learning. It is possible, it's validated, and especially the decision trees algorithm are very good. Especially we recommend out of all this validation and test the generic model using random forest on all the data. It had good classification results, even on replicates. It generalized very well, even on unseen data, and is able to detect outliers, for example, in these RNA-seq experiments. As output of this work, you will find more details in the article. You will find interactive content on this URL. You can check this URL, and we have a GitHub repository where you can install, it's a Python code, and also if you want to use it to generate quality features, you need basically Linux, but we also provide Docker containers. And for example, then after installation, in 2 commands, you will first here generate the quality features for 1 file and run the classification to get this probability. So I want to thank the authors, collaborators, and the University of Mainz. Thank you very much, and I'm happy to answer your questions.
Thank you, Jean. So we have time for a few questions, and we do have questions. Oh, a lot of questions. I'll take the first one. So Luciana is asking, saying, nice talk, thanks. What should be the minimal database size to training the ML model?
The minimum database size?
For training the machine learning model.
Yeah, so we have used different sizes, even relatively small, and yeah, I cannot tell really about a minimum. So in general, the more the better, but from 40, 50, it was already working. But then with this small amount, you are not sure if it will generalize to external data. So this can work eventually with low number of samples on your data, internal data, everything following the same protocol. And to generalize, maybe more data, yeah.
So if the data set size is too small, you recommend using like pre-trained models rather than training our own?
Yeah, so we have models. This generic model is very, very good. It was better than a model based on, for example, ChIP-seq human paired end files. It would better generalize the generic model than this smaller model that has already quite a good amount of files, but it was really specific to one type, and applying it to the same type, but in external data, was working not so good as the generic model that is trained with everything basically, and maybe it's smart and taking some pattern that you cannot imagine. So, but still, we have seen that our model are only trained for human and mouse, and these few data types. So I cannot really tell if other species and other data types will be working. But it was working in the paper we show. You take a human model, it can already be applied to a mouse model in some times. Yeah?
Okay, we still have a lot of questions and very little time.
Okay, I will try to make a short answer.
So one which I think you should be able to answer easily or fast. Elias is asking, since you're using random forest, is it possible to extract the features that led to the classification for troubleshooting?
Yes, we did not do, yes, we did it in another paper, yeah. Yeah, we did it in another paper. So the mapping statistics are very powerful in this set of features. So the bow tie, number of uniquely mapped reads, overall mapping statistics, and the FastQC also coming very strong. And the two other, this location and TSS, are maybe useful, but more for specific subsets. Yeah.
Okay, we will take a few more questions, but people reading the questions, don't hesitate to upvote the question you want addressed, because we won't have time for every question, and you can help me choose the ones that I forward to Jean. I'll just take the oldest one. So Jules Gillet is saying, thank you for your presentation. In the first part, you've shown AI classifier provide better prediction of quality class and manual annotation. The classifiers are trained on annotated data sets from ENCODE, but what are the predicted classes been compared?
So I'm showing it's, yeah, so the manual annotation is not to compare with the machine learning. It's using, machine learning is using it to train and to know how to do the same. And what I compared is you take the machine learning, and if you don't take the machine learning, you just take one of these quality features. Let's say the number of uniquely mapped reads. You compute this value for all files, and you see if these values is allowing you to classify the manual, like the manual annotation. So this is what I compared the single values against all the values combined with machine learning.
C'est ce qui me semblait. One upvoted question here is Julien Fouret saying, thanks for the talk. Did you try to generate models specialized only at the data type, either RNA-seq, DNA-seq, or at the species level? I think you showed that, but.
Yeah, so the different subsets or specialization were for chip-seq, human, and single-hand, chip-seq, mouse, single-hand, DNA-seq, paired hand, etc. So we were at this level of subsetting the data. So it was working. Most of them, these models would also generalize to external data, a few not.
Okay, I'm sorry, but I think we have to move to the next talk. You still have several questions in the chat, which you can read, and maybe people can just join you in meeting rooms in the, what's it called, Gather Town thing, and chat right after this session.
Yeah, or send me an email, or maybe I will try to answer in the thread. Thank you very much.
Thanks for your talk.